So what's the whole point of the action potential? So now that we have this traveling change in voltage, we're knocking down all these dominoes, we're opening up all of these voltage-gated channels all along the whole entire cell membrane of the muscle fiber. Why? What's the point? Well, the point is that action potentials cause the release of calcium. Calcium is necessary for contraction. Releasing calcium requires an action potential. It requires that traveling change in voltage along the cell membrane. Action potentials are needed because the voltage from the action potential is what's going to cause the release of the calcium from the sarcoplasmic reticulum. Those are the storage sacs for calcium. So let's take a closer look at this process. So when an action potential travels all along the sarcolemma, this includes the T-tubules, which are places where the sarcolemma dips down and goes deep into the muscle fiber. Don't forget, T-tubules are just made of sarcolemma. So when the sarcolemma experiences an action potential, so do the T-tubules. Well, the T-tubules are located right next door to the sarcoplasmic reticulum, right? As a matter of fact, we have some special names for these areas where the T-tubules come close to the sarcoplasmic reticulum, right? We call this a triad, and it includes the terminal cisternae of the sarcoplasmic reticulum being located just next door to the T-tubules. Now, the membrane of the sarcoplasmic reticulum is actually connected physically to the T-tubule membrane, right? The T-tubule contains a voltage-sensitive plug for a calcium ion channel. And this plug is plugging up an ion channel that's embedded in the membrane of the sarcoplasmic reticulum. Let's take a look at what this would look like, okay? So down on the bottom, we can see two pictures. One is what the situation looks like at rest, and one is what it looks like when the cell is experiencing action potential, right? So let's take a look at the at-rest picture. We can see the sarcoplasmic reticulum labeled SR. This is our storage sac filled with calcium, okay? We can see the sarcoplasm, which is the cytoplasm of the muscle fiber, right? And the dark orange kind of line is the sarcolemma, okay? The dark blue line that's surrounding the sarcoplasmic reticulum is the cell membrane of the sarcoplasmic reticulum. The purple cylinder is going to be our calcium ion channel, and the pink sort of rectangle plugging it is that voltage sensitive plug that I mentioned, right? So what's going to happen is that when the T-tubule membrane experiences an action potential, and I'll switch to the action potential picture, right? That voltage sensitive plug, that pink rectangle, is going to be pulled out of that calcium ion channel. And then calcium ions will then be free to leak out of the sarcoplasmic reticulum into the general sarcoplasm of the muscle fiber. So let's take a look at this process in an animated form, okay? So when the action potential begins to travel all along the sarcolemma, it travels down the T-tubule sarcolemmas, pulling that plug, that voltage-sensitive plug, out of the calcium ion channels, right? So now the calcium ions are free to flow out of the sarcoplasmic reticulum into the sarcoplasm. They just diffuse. They go from an area of high concentration. The area of high concentration was inside the SR to the area of low concentration, which is in the sarcoplasm itself. So now we have the calcium ions that are required for contraction to occur. Why do we need these calcium ions? Right? Calcium ions cause exposure of binding sites on actin. Once the calcium ions are in the sarcoplasm, the calcium ions bind to troponin. This binding bumps troponin just a little bit, causing the troponin to move. When the troponin moves a little bit, it moves its attached tropomyosin too. When the tropomyosin moves, it reveals the binding sites on actin. So now let's take a look at the process of where myosin actually can bind onto actin and pull. This is the actual contraction event here. Okay? So there's a very strong attraction between actin and myosin. Okay? Those binding sites on actin have a very, 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 very strong attraction to the myosin heads. So once the binding site is revealed, the myosin heads just will automatically pop up and grab onto it. This process does not actually require any energy. This is actually an electrostatic interaction between the myosin head and the binding site. An electrostatic interaction is the fancy way to say you have positives attracting to negatives. You can kind of think of it this way. Think of the binding site on actin as being coated with a bunch of plus charges, and think of the myosin head as being coated with minus charges. So that once the tropomyosin is pulled away, the pluses and the minuses can just come together now. Once the myosin grabs onto the actin, we say the cross bridge has formed. Okay? So once the cross bridge forms, the myosin can now pull onto actin. Right? And these are the steps that are going to occur in this process, but let's take a look at this in pictures as we look at the steps. So first of all, myosin has to bind to actin. We already kind of covered that, right? It doesn't require any energy. The myosin heads just automatically grab onto actin due to an electrostatic interaction. Now notice here in the picture the myosin is bound to the actin. Well, let's look at what's on the myosin head. So yeah, the myosin head is bound to actin, but it's also bound to two other things, ADP and phosphate. And these are important, right? Because what's going to happen is we're going to switch up what's attached to the myosin head, and that's going to allow different things to occur, okay? So right now, myosin is bound to actin, okay? What happens next is the phosphate group is released from the myosin head. What actually causes that to occur is when the myosin grabs onto the actin, that kicks off that phosphate group, okay? And the phosphate group was doing a really important job, okay? The phosphate group was holding the myosin in this weird twisted torque position. The phosphate group was like a pin holding the phosphate in that high energy position. And once you pull out the pin, the myosin head is capable of unwinding or untwisting, and as it does so, it pulls on the actin, causing the power stroke. This is the actual sliding event that occurs during contraction. So the phosphate group was like a dam holding back water. Once you remove the dam, the water's going to flow. So the phosphate was a pin holding the myosin head in a twisted high energy position. And once you pull out that pin, the myosin untwists, pulling on the actin. Okay? After the power stroke occurs, ADP is then released from the myosin head. Right? So phosphate falls off, myosin pulls on actin, and then ADP falls off. The next thing that has to happen is we have to break the cross bridge. We have to have myosin let go of actin. And this is necessary so that this myosin can grab onto another actin and pull. So in order to get myosin to let go of actin, we need ATP. And this is because the attraction between actin and myosin is very, very strong. And if we want myosin to let go of actin, ATP has to come in and bind to the myosin head. And once ATP binds to the myosin head, the cross bridge will break between actin and myosin. And now the next step can occur, which is the repositioning of the myosin head. We have to get that myosin head back into that weird twisted torque state. Okay? So in order to do that, we need energy. And we're going to get this energy through ATP hydrolysis. We're going to break the ATP down into ADP and phosphate. And there will be energy released during that process, and that energy will be used to twist the myosin head back into its torqued high energy position. At this point now, we're free to go back to the first step, which is myosin can grab onto actin, and we can repeat all of these steps. And we continue this process where we repeat these steps over and over again until we run out of something like space. Right? If the sarcomere gets as short as it possibly can, we run out of space and we can't do it anymore. Or maybe we run out of calcium, or maybe we run out of ATP. So we keep doing this until we run out of something, maybe the nervous system signal, whatever it might be. Now, let's not forget the big picture here. Right? The whole point of muscle contraction is to move bones. Right? The process of myosin pulling on actin will move our bones. Right? When myosin pulls on actin, the actin moves. And don't forget, Actin is connected to the costomeres, and the costomeres are connected to the sarcolemma and the endomysium. And the endomysium is connected to our tendons, and our tendons are connected to our bones. So when the actin of the thin filament moves, we can move our bones.